the moderator to this session. Before we start, please take note of these housekeeping rules. Please ensure that your researched and invented multiple diagnostic kits, such as the rapid detection kit for methicillin-resistant Staphylococcus aureus, which has been patented and commercialized since 2015, the multiplex PCR rapid detection kit for hemorrhagic bacteria from stool samples, which he has filed for patent last year, just to name a few. These diagnostic test kits have won him numerous national and international awards in the Innovation Invention competition. Dr. Hassanain has published more than 60 scientific journal articles with more than 600 citations, 2 book chapters, and 3 books. He has also been the recipient of numerous research grants from local and international funding agencies. Without further delay, I would like to invite Dr. Hassanain. Dr. Hassanain, the floor is yours.
Thank you, IP Dr. Wang Somoy for introduction, and it's my pleasure and my thanks to Research Nexus UiTM to give me this opportunity to present and share my products and my research among them. And thank you very much. I hope this presentation will help lecturers in the field of diagnostic microbiology and improve their skill and help them in their future work. My name is Dr. Hassanain Al-Talib. I'm from Medical Microbiology and Parasitology Department, Faculty of Medicine, UiTM. And I'm proud to be one of the a member of our research group in our faculty. Today, I will share with you my products which I have worked on in previously, and the outcome is development of a 3rd and 4th one in the way, diagnostic kits. And in this presentation, this is the content of my presentation. I will just share with you our research group team, and then I will show you my products. Then I will choose one kit to go in more details, and will share with you some technical points and challenges that we faced during the development of this kit. And also, I will share with you why we did this study and materials, methodology, what are the important results and the novelty of that particular kit. Then we come out to the conclusion and achievements. Let me share with you our diagnostic research group. This is our RG team, which is now headed by IP Dr. Wang Somoy, and also IP Dr. Jamal Hussaini, also Dr. Nurul Azira Muhammad Shah, Dr. Chua Anglim, and IP Dr. Edi Hasrul from Faculty of Dentistry, Dr. Siti Farah Alwani, and IP Dr. Yu Chong King. Okay. These are my products or kits that I have worked on during my research journey. So we see in number 1, this kit was developed for detection of important bacteria, Staphylococcus aureus, and which is resistant to methicillin and vancomycin. This kit helps us to detect this bacteria, which is very important to cause a serious diseases, especially among newborn baby. It causes meningitis and septicemia among neonate, and also it can cause necrotizing pneumonia among elder age group. So we developed this kit, and it's already commercialized and patented. Then after that, I try my luck to develop a kit, but now on development of protozoa, parasite, not bacteria. And for us, bacteria is much easier to deal with. Bacteria is much easier than to deal with protozoa and parasite. And this kit in number 2 is used to develop to detect protozoa which cause diarrhea among people. Our third kit, which is just developed last time, this is for detection of also bacteria which cause bloody diarrhea. And fortunately, this kit also now filing for patent, and we achieve, alhamdulillah, many gold medal, and we participated and invited to represent UiTM in many international exhibition. Then number 4, before I go to number 4, so the first 3 kits for detection of bacteria, as in number 1 and number 3, number 2 in detection of protozoa parasite. Then we want to take the challenge and develop a new kit for detection of viruses. And my role in the kit number 4 is a co-researcher, and now our students, actually this project headed by our moderator, IP Dr. Wang and I am a co-researcher, and our student is working on this kit currently for detection of viruses that cause encephalitis. So we will see. Hopefully, we will the boxes will become 4 in the future. Okay. After we got appointment letter to start our RIG group that time, then the university opened a new grant, REI grant, and we applied. I applied for this grant, and alhamdulillah, I obtained the REI with 32K, and we start to work on in the development of this kit for detection of protozoa that cause diarrhea. So here I will go in more details in this presentation about this kit as example of the other kits. So let me ask, is diarrhea as a condition or as a presentation from the patient, is it worthy to develop diagnostic kit for diarrhea? I think the answer, you will find it here. In 2015, diarrhea killed 9% of children, and this is all over the world. And in Malaysia, it killed 3% among the cases. 3% death due to diarrhea. This is not from me. This is from WHO. Now, when we have a case with diarrhea, the simplest and easiest method for detection of the causative agent of this diarrhea by microscopical examination. But unfortunately, microscopical examination characterized by low detection limit and low sensitivity. This is because in microscopical examination, there is an examiner, technician, who look to the field and a lot of variation. Many variation might resulted and resulted in low detection limit. Okay. Microscopical examination depend on direct observation of protozoa by microscopy by the technician. Although this method is rapid and simple, I mean the microscopical examination, but there are many variations. This variation could be due to the skills. Maybe the examiners of this slide of this specimen are not skilled or not efficient to find the causative organism. This is number one. Number two, the sample. If the sample is fresh or late, this might affect the result and the finding of the result. Also, let us say the person can identify the organism, but for protozoa, there are similarity between pathogenic and non-pathogenic protozoa. Or in another way, we have two species. For example, Entamoeba histolytica is pathogenic. Entamoeba coli, non-pathogenic, meaning it not cause symptoms. So morphologically, both these two species by observation looks similar. So here the examiners might be confused and give wrong diagnosis, and the result will affect the patient in the later on. Another reason, some protozoa that included in this study cannot be identified by the technique or the staining technique used by the technician. You know, when we receive stool sample, we use a technique or stain by direct iodine stain. This method cannot detect one of the protozoa that we included in this study, and this is Cryptosporidium. Cryptosporidium need special stain, which is called Ziehl-Neelsen stain. And during the daily lab work, the staff or the technician usually do direct iodine stain. That's why this Cryptosporidium will be not observed, will not be seen due to this reason. Okay, one more reason resulted in variation in microscopical examination is the method used in the preparation of the slide. If the technician used just direct method by direct preparation of the slide, this will result in low sensitivity in comparison to the concentrated method. By using concentrated method, it will give high chance, or the sensitivity will be high for more detection. But we cannot guarantee that all laboratories in hospitals using the concentration method. That's why when we compare microscopical method with the molecular method, we can say that molecular detection or detection by molecular method is simple. Maybe some argue with me and tell me PCR is not simple method. It needs multiple steps for preparation of the master mix, for loading of the gel. But I can say that this by time will be routine procedure. and the technician can make it as a routine procedure. So easily to learn, not that much difficult. And also, I want to say that molecular detection method also offer a better turned around time. Also here, I would like to explain. At the beginning, I said that microscopical method is rapid and simple. For PCR, it need extraction, it need preparation of the master mix, loading of the gel. But let me explain this, please. For microscopical examination, when we prepare the slide, it should be examined on the same time. Because if we left the slide, it will dry and it will not give accurate result. That's why the technician should be engaged in each sample one by one at different time. While in molecular method or by PCR method, we can deal with a batch of samples, 10 samples, 10 samples, or even 15 samples at the same time. The process came out in the same time. That's why it will be much easier, and by calculation of time, this will be at the end result by PCR will be less time, will require less time. Now I come back to our PCR. In our PCR, we used 3 pairs of primer for detection of 3 important protozoa and +1. I didn't say 4, I said 3 + 1 because this one is specific primers for internal control, for validity of our kit, to show us our kit is working or not working. And I will go in more detail later for the internal control. So in our study, the aim of our study to develop and evaluate a molecular diagnostic kit by PCR, and this kit will be able to identify Entamoeba histolytica and Giardia lamblia and Cryptosporidium parva. So these are the 3 important protozoa which included in our kit. Let me just go and elaborate on the background of our study and its relation. Our kit, our developed kit, is a modified and inclusive method which can identify any of these 3 protozoa simultaneously. What does it mean? It means that if we have patient sample, or we have stool sample which came from endemic area, and he had the 3 protozoa or 3 pathogen at the same time, our kit can detect the 3 pathogen on the same time. If the patient presented with 2 protozoa, our kit able to give us 2 bands and tell us that this patient infected with 2 different protozoa. Same thing if 1. So it can detect any of the 3 pathogen alone or in combination. We compared our result in the lab with the conventional method by using third party, and the results were matched between our finding and the conventional methods. We include internal amplification control. We include internal control in our PCR kit, and this is what I said, +1. Internal control, why we put internal control in the PCR? If we look to the literature and those who work on the PCR, we found most of them didn't include this internal control. And during my research time, my advice to anyone who work on PCR to include internal control. Internal control can detect PCR inhibitors. We have some biological samples, and these biological samples contain materials that resulted in inhibition of the whole PCR amplification. And at the end, it will not give us band or amplification, and we will consider this is negative. But it might be false negative result due to presence of PCR inhibitors. So also this our study will help to incorporate DNA amplification technology in the diagnosis of parasitology laboratory to detect the causative pathogen. As you know, currently the new technology depend on the molecular methods, and during pandemic due to COVID-19, we can notice clearly that health and medical authority trusted only the PCR. They didn't, I mean, they didn't depend on rapid detection kit for suspected. So, what are the reasons that make us to develop this kit? A large proportion of diarrhea illness are ascribed to unknown etiology. And I explained the reason earlier due to wrong diagnosis. So the result come out, no pathogen was detected, no oocyst. But actually, this might be not correct result. Molecular methods replaced the conventional methods due to rapidity and high validity, especially after COVID-19 pandemic. Early diagnosis is very important and enable evidence-based intervention, reducing morbidity and mortality. Of course, early diagnosis early diagnosis resulted in early treatment and result also leading to less spreading of infection among people, and of course this result in lower morbidity and mortality. What about previous molecular methods? There are many diagnostic kits available in the market, but when we search in the net, we found that majority of these kits dealing with pathogen individually. So, meaning they develop kit for Entamoeba histolytica only, or they develop kit for Giardia only, and same thing applicable to Cryptosporidium. But our kit combine the three pathogen in one kit in one processing. Other kits need to process one by one. This multiple pipetting and dealing with pathogen might result in error. sRNA. Most important part in the development of the kit is the designing of the primer. Usually I advise my students to do it very carefully, and I do it with them because we should design primer to be specific 100%, and we did the blast many times and confirm that our primer matched with the gene sequence that we want to amplify, and not cross-react or not amplify other gene sequence from other bacterium or other gene. Then also, when I design my primer, I designed manually. I prefer to design my manually because I want to choose the product size of my base pair, the base pair of my band. So there will be a clear distance between band and band. And this will make the reading of the result easier and more accurate. And the melting temperature of our combined primers was 70 degrees. For DNA extraction, we use the Kaya stool mini kit for DNA extraction, but when we follow the standard protocol of the kit, the amount of DNA very little. So we did modification based on reading of our literature. So those who want to work on protozoa, you need to do some modification. You cannot obtain good amount of DNA when you follow protocol. That's why we boil our oocysts and for 100 degrees. For 10 minutes, then we also use proteinase K for 2 hours at 55 degrees. This is very important, and if you compare the amount of DNA, will be more yield with this modification. Then we proceed for optimization, and we should go through optimization of single primers and gene uniplex, and then after we achieve the result, we proceed for the multiplex optimization. Also, we did optimization for arenic temperature and the primer concentration. Why we did optimization? To obtain a uniform and clear bands for all target genes. This is our aim of optimization. Also, to avoid effect of one primer on the other primer. I mean, not to one being overlapped by other. So that's why we do optimization. And after amplification, we load 6 microliter of the PCR product, and then electrophoresed using 1% agarose gel containing cybersafe DNA gel stain. After that, we did analytical sensitivity and specificity. For analytical sensitivity, we use various concentration of genomic DNA, starting from 50 nanogram to 600 nanogram. And this is to know the lower detection limit of our kit. Then after that, we want to confirm that our kit not interfere with other organism and didn't give us wrong band. That's why we conduct analytical specificity using 11 strain, and including our target protozoa, in addition to other 5 non-protozoa bacteria and other 3 related protozoa. Now, in the result, when we starting our optimization process, this is the result. There is no any band. we couldn't obtain any PCR amplification product when we follow a standard protocol for PCR. And this take long time to solve this problem. By the way, the whole process for from extraction until we got the band take about 5 hours. So this is in terms of time. I come back here. So by following the standard technique, we couldn't obtain any band. So we need to do more and more optimization and look to the reason. Then by referring to literature, we found that using DMSO 5% improve our PCR, and we managed to get a clear band, as you can see here. This is from our gel electrophoresis. Here in this, I will show you here. This is the band for Campylobacter. It's about 112 base pair. There is about 200 base pair difference among the next target gene for Entamoeba histolytica, which is 300 something, and then the third one, Giardia lamblia, 500 something. So DMSO improve our PCR because we found that DMSO reduce the secondary structures that affect in the PCR and it enhance amplification, and DMS also relax the supercoiled plasmid for the DNA. So I recommend those who's working on PCR with not clear bands or no bands to use DMSO and compare the results. The other challenge that we find during our development of the kit, this is for Entamoeba histolytica primers, which fail to give us a specific one band. And if you can see if you can see this gel electrophoresis, our target is enclosed in the yellow line, while we found that there are another band appear in 200 plus something. These bands, which is surrounded by the red lines, are non-specific. So there is a non-specific amplification of Entamoeba histolytica. So why these two bands appear? Now, if you count the bands, there will be 5 bands, and it's supposed to be only 4 bands. So after search, we concluded that our primers that we designed can bind to another site in the gene, but this binding site in the gene not updated in the gene bank or not published on the gene bank. That's why when we did the blast, cannot capture by a gene bank. So what we did to overcome this problem, we did more optimization by reducing primer concentration and increasing annealing temperature. So fortunately, this non-specific band disappear. Then for analytical sensitivity, we found that our PCR can detect the DNA with 50 nanogram. The lowest detection limit of our PCR is 50 nanogram. For analytical specificity, this gel electrophoresis showed that there are no bands by using other bacteria from GIT and other protozoa. So there are no any bands for this. This band, all the bands on the top, this is belong to internal control. And let me say here, the internal control band is similar to what we can see in RTK, rapid detection kit, the strip one. When we did the rapid detection kit, and we can see only one band. So that band is considered as a control. If there is two band, meaning the person got positive COVID. So, but if one band only, this is considered negative. So here, this band in our gel electrophoresis is similar in action or similar in function to that band in RTK. And it tells us that the whole PCR condition is working perfect. Because as you know, in PCR, during PCR work, we have, many variation. Something related to the program of the thermocycler, maybe the hot plate not working properly, and other factor related to the master mix preparation. So presence of this internal control band indicate that all conditions are perfect for PCR to be amplified. If we look to LAN under 12, this is our positive control. LAN, which show us the internal control Giardia, Entamoeba, and Cryptosporidium for comparison of our sample with this LAN. What are the novelty in our kits? Number one, direct detection. So we can use direct stool and immediately proceed for extraction of DNA and proceed for PCR. So no need for any processing, any concentration, or any incubation. Also, the detection limit is 50 nanogram at the genomic level, which is considered, acceptable for, for the protozoa. Maybe for bacterial, the low detection may be lower. Also, our PCR is rapid and can give result within 4 hours, which is essential for identification of protozoa in the same day. This is the most important, in the same day. Number 3, including of internal control will make our kit trustable, and it can detect any PCR inhibitors. Also, our PCR can detect presence of one or more pathogen simultaneously. In conclusion, the developed PCR assay has a real potential to replace the conventional routine test in diagnosis of microbiology laboratories, especially among countries with high prevalence of enteritis. Also, we might need more work and more research in development of kit which include more pathogen at the same time. And we might include some bacteria and some parasites on the same kit. It comes out with one kit only. This is our achievements for the kit. For the earlier kit, we participated at the national level in IDEX 2018, and we achieved bronze medal. And we come out with the paper, which is published in Index journal, and also we obtain a copyright for our kit. This is our kit and our future plan to proceed for commercialization with industrial companies. So after development of the previous kit, we want to do another kit for detection of bacteria that cause bloody diarrhea. That's why I applied for PRGS, Prototype Research Grant Scheme, and I awarded the grant with 124K. Then this kit, I will just summarize the outcome of this kit. It's already developed in our lab in IMMB and already submitted for filing, a patent filing now with my IPO. For the second kit for detection of hemorrhagic bacteria, we came out with the 3 copyrights, and we published in Q1 Journal of Clinical Microbiology. And also we have industrial collaboration with USM and Medivine. We participated in international exhibition for invention and exhibition competition, and we got gold medal in ICANN in Toronto, Canada, and also we received special award recognition from Helier company from Canada. And also we participated in ITEX International Conference nominated by University in ITEX 21. And also our university nominate our product for MTE 2022, and both we achieved gold medal. And then currently also we already submitted our product for Pechipta. Many thanks to UiTM and RMC to send our product for Pechipta this year. At the national level, we obtained gold medal in the International Medical Innovation, which is organized by our faculty, and we obtained top three among innovation and academician category under medical innovation competition, our faculty also. And also we obtained gold award in IDEX 2021. In summary, the outcome of these kits is two patents and maybe 6 or 7 copyrights, and we publish 3 papers in Index and Q1 journals. Many thanks for your listening, and many thanks for UiTM and our faculty for their support, and ready for your question. Thank you very much.
Right. Thank you, Dr. Hassanain, for the very informative presentation. Now I would like to open the floor for question and answer. Are there any questions from the audience? You can unmute and ask a question directly, or okay, I think there's one question in the chat. Okay, Dr. Hassanain, there's one question in the chat. Okay, the question is by Nora Noazima. Okay. Assalamualaikum, Dr. Hassanain. Do we need to incorporate internal control to run PCR for known positive samples?
Okay, thank you very much for this question. Although during the presentation, I give advice to everyone who conducted PCR to include internal control. As you know, PCR is different from real-time PCR. The PCR either positive or negative. So it is qualitative, not qualitative. Yes or no, positive or negative. So presence of internal control, because for internal control, you will include everything, the primers and the target gene within your master mix. So it will be distributed and mixed with your sample. When you run the PCR, it can tell you, presence of the internal control band will tell you that the whole PCR condition is perfect, working well. During my experience, my RA came and bring me result. When we look to the result, at the beginning, I look to the internal control band. If there is no internal control band, meaning that there is either PCR inhibitors, which inhibit the whole PCR, no amplification, or might be something related to the thermal cycler. When we compare between different machine, or we change environment, change the condition, PCR condition, and obtain internal control, then our reading will be accurate 100%. Nobody can argue with you. Thank you.
Okay, Dr. Hassanain, maybe can I add on? What type of gene you can use as an internal control? Can we use a human housekeeping gene?
Okay.
But this one is for protozoa, right? Yeah.
This is for protozoa. I use cloned gene from Vibrio cholera. So I just took the target gene and I designed a primer for this. So I tried to choose some non-related pathogen, but any gene sequence with primers will act well.
All right.
Next, Doctor.
Okay.
Okay. May I ask a question? How do you ensure that your primer is very specific? Because for me, I only blast the sequence of the primer using the blast nucleotide, and I check the primer sequence on the Mega software. So, do you have any other software that you use in order to check the primer specificity, Doctor?
I use BLAST, and to me, BLAST is very informative and give you the percentage of similarity and list down the bacteria or related organism which have similar. So to me, I think BLAST is okay. BLAST is okay.
Okay, thank you, Doctor. And another question is, I am not really familiar with the internal control. So, how do you create that internal? How do you create that internal control? Is it like how you design the primer?
Okay, as I told you, for me, I use a cloned gene from Vibrio cholera that is previously developed during my PhD study in USM. So that particular gene, the cloned one, we design primers on this gene. So now you have the target, gene, and you have primers for this. These two components added to your master mix, and it will be distributed on your all samples. So all your samples will contain the target gene and primers. So the band will appear on all samples.
I see, understand, Doctor. So, if I already have the positive control template, so I don't need that the internal control, right, Doctor? Is that right?
Again, repeat again, please. If you have.
So if I have the positive control template, which already have the target gene sequence, right? So I don't need that internal control primers, right? To incorporate into my master mix.
Okay. Positive control, your positive control, you add in one sample only, correct or no?
Yes.
So in this case, your positive control will be from reference strain, like ATCC or very known strain, and the DNA will be, the concentration will be high, and you will compare your samples, you will compare the bands of your samples with the positive control. But I come back to your question. Your positive control will be only in one lane, correct or no?
Yes.
So it will not be present on all samples.
I see.
While for internal control, it will present in all samples. It will be an extra band in your sample. You got the point?
Yeah.
So in conclusion, positive control cannot replace internal control.
All right. Thank you, Doctor. And my last question is, I have one pathogen that cannot be amplified using the real-time PCR. So can I use, because previously you mentioned that you use the MSO, right? When you encounter the problem of not getting the band when you conduct the gel. So can I use the DMSO?
Sure.
Yeah.
Try to add 0.5 DMSO to your master mix, and hopefully you will get.
Yeah.
Hopefully, will improve your result.
Yeah.
And if not, can contact me. Could be later. I mean, if I can help you.
Yeah. Okay. Thank you so much, Dr. Hassanain.
Welcome.
Yeah.
Are there any questions from the audience? Let me check. Check the chat group. Okay, perhaps I can ask the last question, Dr. Hassanain. Just now, the kit that you described, right, is a conventional PCR kit, right? Do you have any plan to like convert it into a real-time PCR or LAMP?
Yes, that is. I mean, the future plan is to use real-time PCR. In real-time PCR, as you know, Prof, it will be quantitative and qualitative, number one. And number two, it is more advanced to the basic PCR, and no need to use the ethidium bromide or this non-harmful stain. And the most important things currently for commercialization, if you are dealing with real-time PCR, will be more acceptable and more marketing will be more than the basic PCR. So yes, there is attempt, there is a plan for the future work to be all on real time PCR based.
All right. So I think are there any more questions? Let me check the chat. There are no more questions. Since the time is almost 11, I would like to thank Dr. Hassanain again for willing to be a speaker for this webinar series. So I hope everyone benefited from Dr. Hassanain's talk. So before I end, may I remind everyone to fill in the feedback form. Okay, your suggestions and comments are very much appreciated. All right, so thank you everyone. See you next time.